All right. With that, Marshall McDaniel, assistant professor in agronomy, is with us today. He is an expert in soil health, and I know a few people, a couple who couldn't make it today, are really excited to learn more about soil health and manure. So with that, Marshall, we look forward to hearing about soil health and some of your ideas and things that you've done. So thanks for joining us today.
Thanks for inviting me, Dan. I appreciate it. Yeah, I'm really excited to chat with you all, and I want this to be informal as possible. In fact, I'll have some pauses for questions, and so feel free to chime in, raise your hand, just shout out if you have a question about anything. As Dan says, it's not in my official title, but I have embraced the soil health movement and jargon. It was kind of research I was already doing, but now my research path is kind of going that direction because there's a lot of interest there, both grower interest and interest from research standpoint. So I'll kind of fall somewhere in the middle there, and I'm going to talk about some heavy science on some soil health measurements, and then some kind of easier, and my research program has actually been going this way, some easier do-it-yourself soil health tests. So I'll give you a little bit of flavor of both of those ends of the extreme. But I told Dan I wanted to keep with the alliteration. So not only is my name also 2 M's, but my title of the talk is, Are Your Mollisols Magnificent or Marginal? And I thought this was a good way to frame the idea of soil health, and really we want to determine whether or not we're making improvements with the change in management, or we're staying at status quo, or what I would call marginal mollisol. All right, so let's get started. Can everybody see my laser pointer too on there?
Yes, it looks perfect.
Okay, great. And this is actually a picture of a real soil. This soil on the right here is from a 12-year-old prairie reestablishment. So there's roots in there that are from prairie roots. The aggregation, the soil aggregation is actually visibly, noticeably different. And hopefully we'll have time to look at that compared to a corn, soybean rotation with just synthetic fertilizer that I consider our kind of baseline for much of Iowa. So as I go through this, feel free to chime in, as I mentioned, but also feel free to email me if I don't get to your question or you think of a question later. My email is marsh@iastate.edu, and you can follow my lab group on Twitter and all the cool stuff they're doing. It's really not me doing it, but the lab group that's doing all the hard work. And it's soil_plant_IXNS, which is abbreviated form of interactions. And we also have a website where we feature the work as well that a lot of these students are doing. Some of them actually have left the lab already on to other jobs. But feel free to contact me, and I need to give them credit where credit's due. They're doing all the hard work. I just get the easy job of chatting with you all about it. Okay, so with soil health, I always like to start with this, both in the classroom, talks in front of producers and growers, and actually I use this a lot in my science talks as well to scientific audiences. With soil health, I like to think of soil function. So soil health is really an umbrella term, and it's actually quite vague. It would be the same as if I said to Dan, you know, I improved my health the other day. Dan would want to know more details. He'd want to know, okay, what did you do? You know, what aspects of your health are you talking about? Are you talking about your body mass index? Are you talking about your cholesterol? So with soil health, I like to think of the specific soil functions. And this is a pinwheel from the Food and Agriculture Organization showing those functions. And many of them are related to agronomy, which is the department I'm in at Iowa State. And some of them are not so much, like cultural heritage. Soils actually store cultural artifacts, but this is something of more concern to archaeologists and anthropologists. Some of my colleagues in engineering, they are concerned about it for the Foundation for Human Infrastructure. I couldn't be here in my office if soils didn't support the building that I was in now. But I want to highlight all the ones that I am kind of more in tune with from a day-to-day basis being an agronomist. And this would be provisioning of food and fiber fuel. These are the ones I highlighted in yellow. Carbon sequestration is all the rage now. We could talk about that if we've got time left over. Water purification, soil contaminant reduction, climate regulation related to the carbon sequestration, but now we're talking about the other gases involved in climate change. Nutrient cycling, so this is part of the provisioning food, fiber, and fuel. So some of these soil functions or soil ecosystem services are related. The habitat for the organisms that also drive a lot of these processes like nutrient cycling. Flood regulation is also important, infiltration rates, water storage. But all of these, and this is the other addition I've made to this Food and Agriculture Organization chart, soil organic matter is really central to all of these functions. And it's why I put it kind of here in the hub of this wheel of these functions. Another way to look at that is how do we then improve those functions? So I gave you the list of the things that we might measure under an umbrella of soil health. These are actually the principles, and I borrowed this from the NRCS. This is their 5 principles of soil health. And they've got them listed as soil armor. I think I've changed the naming a little bit. Minimize disturbance, plant diversity, continual live plant root or perenniality, I might refer to it as. So having living roots or living plant longer duration of the year. And then livestock integration, which really hits home in this wheelhouse of this talk. And I'll just go through a couple of the practices that might cover these soil health principles. So cover crops, leaving residue on the surface, using CRP or prairie strips can provide soil armor. Minimizing disturbance, some of these are pretty self-explanatory. Reduce tillage, maybe lowering compaction through controlled traffic. Again, CRP prairie strips might fall under this too. Cover crop mixtures, so cover crop rotations, intercropping, CRP or prairie strips. You know, when you convert to CRP though, you're not getting the food, fiber, and fuel provisioning, but you're getting other soil health or soil ecosystem services. Cover crops again, perennial crops, prairie strips, relay cropping, CRP are a way to have continual live plant root and perenniality. And then last but not least, livestock integration can come in a couple different forms. So there are a lot of growers. Some of you might be grazing on cover crops or grazing in seed pasture rotations, so alfalfa rotation perhaps with grain crops. Or I also consider just adding manure part of livestock integration and filling in this last principle of soil health. So the animals don't need to be there necessarily. There are some proven benefits of actually having the animals kind of feed and tug on the plants, and this might instigate greater root growth and root exudation and have some other benefits, but their manure is a lot of the benefits they add as well. Okay, so now I'm going to shift gears a little bit. That's a little bit of the background on soil health. So what I mean by it, the soil ecosystem services we want to measure, we want to be specific about. The principles that will get you to a healthier soil or magnificent mollisol rather than a marginal one. But here is really where the rubber meets the road, and I like to start talking about these soil health tests because a lot of people have questions about these, a lot of growers I work with. And they want to know, you know, what can I do with the soil test, or which soil test should I use? And I always start by saying, know your goals and objectives. This is important for almost anything you're going to do on your farm. And it's no different with soil health tests. And these are Marshall McDaniel's lists of the purposes of a soil health test, but it might not be the end all or be all. And these are kind of evolving too as these tests are evolving. So the number one, I think, test or the number one reason or goal or objective of these tests is just to show improvement. So going from a marginal mollisol to a magnificent mollisol. You want to show improvement, you want to document evidence. A lot of growers I work with are very scientific minded. They want to show me the data. So to show improvement, you want to show a change in one of those soil ecosystem services after a change in management. So through time. So you might have been doing business as usual management practice, and then let's say you decide to integrate cover crops and livestock, and you want to show a change in one of those soil ecosystem services through time. Or what we do a lot as scientists, where you compare different management in space that you actually start at the same time. So Dan and I are collaborating on experiments. Other folks in my department and his department collaborate in these randomized complete block design experiments where we randomize how we manage the soil. We might have at least 3 replicates or more. We start the management practices in place, and then maybe 5-10 years down the road, we will measure the changes in a soil property or soil ecosystem service. So that's the most common. I've got a green check mark here. The 2 other purposes I'm going to show you, we're not quite there yet, but these are the things you might see on the horizon in the next 2 to 5 to 10 years. So number 2 is maybe a soil health test someday will inform other management practices. Now, I've talked to Dan about this a few times, and the one that really comes to my mind, and there's some evidence for it already that I'm going to show you some data, some synthesized data from, is reducing nitrogen fertilizer rates. So can we use one of these tests that might measure biological activity, for example, to reduce nitrogen rates for a certain field after, we've built up this idea of nitrogen supplying power and potentially mineralizable nitrogen. So I'm going to show you an example of one of those tests in a few more slides, but I'm just going to go to the next one first. We're not there yet. I don't have a slash through it, so we can't do it. But we've got some preliminary evidence that's showing promise, and I'll show you that evidence. The third rationale or goal or objective for using a soil health test might be to increase farm income. And this is where we see a lot of hype around carbon and ecosystem service markets now. So if you can just document changes in carbon, that can add another stream of revenue to farming. And then, and we're definitely not there yet, but this is something I've been talking to my colleague Lee Burris and others have come up with. You are all probably familiar with the CSR2, the corn suitability rating version number 2. So we think that there could be a better version, maybe CSR3, that includes a management factor that's based on soil health variable or measurement. And that would allow people to sell their land at a higher price, because right now, currently, the CSR2 does not allow for a management conservation practices inside that calculation for the CSR2. So that's kind of where we're at with goals and objectives for soil health tests. I'm going to take a pause to drink my tea, and if anybody has any questions on those or what I presented so far, go ahead and go for it.
I have one on the carbon market, or at least an intro to the carbon market. Do you think it's going to be based on documented change in carbon in the soil, or is it more, these are practices we know can help, and you get paid for those, or some combination of both?
That's a great question, Dan. Yeah, so where the market's at now is it seems like we have a lot of validation to do, right? So the science actually is still debating, which practices and where, under what soil texture or environmental variables increase or set your trajectory on carbon accumulation or accrual. We know that we're never going to get to a point where we can sample and verify every single farm. So there'll probably be maybe not exactly what you said, just we know these are the best, management practices to increase soil carbon, but maybe a combination of a little bit of validation, but also modeling too. So some of my colleagues and your colleagues use models to look at these things, and maybe some remote sensing as well to predict changes in carbon. Right now, some of these companies are taking baseline measurements, and either some are using shallow cores and others are using deeper cores, and they're just kind of getting a start, and it's really in nascent stages right now. But I think there'll always be a place for validation or collecting soil samples. So it'll keep me in business. Any other
Thanks.
questions along that line? That's a good one. Okay, everybody's eating their lunch, I guess, too. I would be too. Okay, so I mentioned how I was going to present some more laboratory-based, more research-oriented soil health measurements alongside some do-it-yourself measurements that my lab is trying to coordinate and see if we can use some of these easier DIY measurements as a proxy or to estimate some of these more expensive, more laborious measurements. So I've got a list of a couple of these measurements and then which soil ecosystem service they give us an insight into. So microbial biomass is a common one we use in my lab. What we do is we actually gas all the organisms inside the soil with chloroform. So in the movies, it's the gas you see they put on the rag and knock people out and throw them in the back of the trunk. So we actually can use this gas to explode all of the organisms in a soil, and then we can extract all their guts, their insides. And then with a control, we subtract the two and we can get how much living biomass is in the soil. So that's an actually pretty easy measurement. There aren't a lot of soil testing labs that do that, but that's a measurement we are a big advocate for. And we think it's a proxy for the stability of the soil, how much carbon is accruing in kind of the nascent stages, and then also nutrient storage and release. I'll get to in a little bit. A few more slides. The CO2 burst method is, some of you might know this as part of the Haney test, which is where you wet up a dried soil and measure the amount of CO2 that's released in 24 hours. This gives us a good indicator of nutrient storage or nutrient release. I'll show some data from that test, actually, some pretty compelling data to me. But like I said, we're not at the level yet where we can start making recommendations on this. Jeff, did you have a question? I saw you unmuted and maybe heard something from you. Jeffrey, I should say, not Jeff Pryor. Okay, maybe Jeffrey just accidentally unmuted there. So total carbon or soil organic matter measurements, those give us, it can be a good indicator of water storage capability, soil stability, carbon accrual, that's a direct measurement, right? Not trying to make inferences from. Water holding capacity, so we have an easy DIY way to measure water holding capacity in my lab. This gives you the amount of water a soil can store, also gives you an indicator of the stability of that soil. Another kind of easy DIY test, we used both cotton in the soil. Most of you might know as the soil your undies test. We've also been using tea and popsicle sticks to bury in the soil and measure how fast it decomposes. Usually, at least the premise we're under right now is the faster it decomposes, the more nitrogen availability there is, the more biological activity. There's an aggregate slaking test. There's a DIY measurement of doing that. And there's also a mobile phone app that I'll show you in a moment that you could use to measure that. It gives you a measurement of soil stability. And then, and I tell a lot of growers this, you can actually just go out with a shovel and look at, you want to be the observer of change. You just look at the change you see and smell and use your senses to look at the change. And so the way I like to think of these options, so these are a range of options. Some of them cover, and they're kind of darkened by the number of soil ecosystem services we can make inferences about. So the darker they are, the more inferences they give you. The lighter, they can give you one inference. But they also, I like to plot them on a measurement cost from low to high versus measurement precision and accuracy. And so this is something we're currently doing right now. I've got a student who's about to submit a paper on this as well. We're looking at the precision and accuracy of some of these measurements versus others. And we think of microbial biomass. I mentioned there aren't very many labs that do that. The ones that do, this is a very expensive measurement. It's usually $50 to $75 a sample just for that. The CO2 burst is a little bit more inexpensive. You could get the Solvita paddles that are about $13 a sample. Soil carbon, or SOM, is a relatively inexpensive measurement. This usually comes along with the suite of soil health tests that you get as well. Water holding capacity is pretty inexpensive the way we do it. Same with the aggregate slaking test. Bearing stuff and measuring the decomposition, all you need to do is, buy some strips of cotton and then get an inexpensive balance, usually $30, $40 from Amazon, or relatively inexpensive. And then the senses, using your senses is actually the most inexpensive, but it's also the lowest precision and accuracy, because if Dan and I were to look at a soil, we might see different colors, we might see the structure slightly different. It's a subjective measurement, but at least it's something you could do very inexpensively and observe changes in your soil. Any questions on this or thoughts on these tests right now before I move ahead to some of the data?
I have one about how stable you think some of these tests are across the growing season, or what time of year should I be thinking about some of these tests?
Oh, great. Yeah, that's a great question. I'm glad you brought that up. So most of these tests, some of them are a little bit more stable during the year. Usually physical tests are a bit more stable, although my colleague, Dr. Horton, a soil physicist, would argue that some things like bulk density I thought were relatively stable actually change quite a bit over a growing season, especially if you're introducing tillage. That's going to really throw things off. But things like microbial biomass, the CO2 bursts, there have been studies that do show they fluctuate depending on temperature and moisture. The water holding capacity, that's one of those physical measurements that are more stable through a growing season or over a year. But the most important thing to consider about these tests are a little more variable throughout the year, or unstable, is just to collect, if you're trying to compare two treatments in time or in space, just make sure you collect them both at the same time of year. And that will make it more likely that you're collecting at the same temperature, same soil moisture. It's also good to take measurements, take a turkey thermometer out there with you, get the soil temperature when you collect a sample. If you, for example, if you do a CO2 burst or anything measuring microbial activity or biomass. So that's one way to kind of explain or maybe interpret your data later, and if indeed you collected them at different soil moisture or temperatures from one year to the next. Does that help answer your question, Dan?
Yes, it does. I should think of them a little bit more like late spring nitrate test and interpreting that than maybe phosphorus concentrations in the soil and interpreting that for a lot of them is what I got. So I think
Yeah.
that's helpful for me in trying to understand what they mean and what I want to try and do with them eventually.
Yeah, you're right. Yeah, so these tests, that's a good analogy. So these are like late spring nitrate tests. They're going to vary. We know nitrate declines over the growing season. There is usually like a pattern with microbial biomass. It goes up, peaks at about July or August. Actually starts to go down in August because it's warm still, but it's too dry for many microbes. And then it might come back up in the fall, especially if we have a warm fall. CO2 burst usually follows kind of a similar pattern. Decomposition. So the most important thing, again, just to emphasize that, is if you do want to do one of these tests, make sure you measure at the same time of the year, because your chances of the conditions being similar are more likely. And these are all the DIY measurements. These are measurements that labs will do for different amounts of money. These are ones that are relatively inexpensive you could do on your own. And if you are interested in doing one of these tests, email me and I can send you protocols and where to buy the materials. Okay, let's, I already mentioned this. I like to rank this. These are the senses. So as I mentioned, this is a subjective test, but it's actually a really good test, especially if it's you that's monitoring it from year to year. Yeah, we lose our vision a little bit year to year, but if you want to observe changes in a management practice, vision is usually the best way to do it. Touch might be the second, so how friable your soil is or how easily it breaks or how moist it is, smell. Actually, many of you have heard of the smell that actinomycetes create in the soil. So they produce this compound called geosmins that gives soil that earthy smell. So you might actually notice a different smell between doing a conservation management practice like adding manure or integrating livestock on your farm and then coming back out to observe these changes. You might notice good smell, we were talking about bad, what was worse, either baby poop or animal poop. And yeah, Dan said so far it's the animal poop. The next thing that's probably the least useful of all of these senses is your hearing. So there's not much you can hear in a soil, although there is a really cool project I just found out about on Twitter a couple of months ago where people are burying these high sensitivity microphones in soils around the world. And you can actually click on a soil in the world and hear it. And it sounds cooler than it actually is. When you click on each one, it just sounds staticky and you don't hear much, but the idea is cool. And then the final one, I'll let you all determine whether or not you'd like to taste. So I challenge my students in my class to taste soil. I'll still taste the soil, but you could use taste to determine the difference between different management practices. But there are a lot of people that are a little icked out about eating soil. Okay, so I'm going to talk about a couple of these different tests now. I'm going to talk about one in particular that's a DIY test, and mobile phones have really allowed us to do a lot of these measurements with the touch of our phone and maybe with a little bit of additional hardware too. So there's a couple benefits to this. One, it's because these measurements can be easily done by anyone. You don't have to send your soils to a lab. And if you're contributing to a larger data set, you might contribute to the broader science and our scientific understanding of how management practices affect soil health. So these aren't by any means an exhaustive list, but these are a couple apps that allow you to do this. So this tea bag index uses the decomposition of tea I mentioned earlier as one of the DIY soil health tests. A group in Canada came up with this worm tracker. This is to track invasive worms, but it allows you to get some information on worms and how to collect and ID some earthworms. This microbiometer, they actually can measure a proxy to microbial biomass, but you have to pay a couple $100s for a kit and some reagents, but that's a pretty useful test. This one I'm really excited about because it's inexpensive other than just paying for the sensor, but it uses a very objective sensor rather than me and Dan using our eyes and arguing whether it's dark brown or light brown. This sensor actually gives you the RGB values, red, green, blue values of a soil, and it has an internal light source to control or normalize for any variation in light that you might have around you. So you place this over a dried soil and it gives you a brownness color. So this could be used as a way to measure your own organic matter. And if I remember right, I think these sensors are just maybe $100 or so. And then the app, I think, comes free with the sensor, this mixed color sensor. Has anyone used one of these before? You can use it for paint too, to get the exact paint color you need for your house. I guess not. No one's used that.
I haven't, but now I want one for all sorts of things.
I know it seems like there could be a lot of uses for it. Especially, I want to try it out just for this organic matter measurement. But
I'm going to use it to match stain colors on my wood.
Ah, yeah. There you go. There's other, so those are ways to get biological activity or organic matter. There's some apps that give you general soil information. So many of them are interfaced with the Web Soil Survey or Surgo database. Land PKS and Soil Web are both good apps I'd recommend. And then there's a couple apps that give you physical properties. So soil structure, this visualize assessment of soil structure. All you need with that is a white tarp or a white tray, and you can go out and you you place a shovel full of soil onto this, and then you make some observations. It's a little bit more qualitative than quantitative, so it won't give you a number, but you can estimate your visual structure, so it's a little bit moving beyond observed senses. And then the Slakes app is really cool, and that's the one I'm going to show a little bit more detail on. This was developed by some colleagues in Australia when I was a postdoc out there. So the way the Slakes apps work, so it used to be only for Google phones, but now it's open for those of us that have iPhones too. So the way this works is you have to get your iPhone and you have to find something to hold it up above a Petri dish and a white piece of paper. That plastic Petri dish you fill with water, and then you drop the aggregates inside. And then the software, what the app does is it uses image analysis, and you press go, and it measures the diameter of your aggregate as over time, as it starts to dissolve and into the water, and gives you a stability index based on the rate at which that aggregate dissolves. And so the faster that your aggregate dissolves, the more unstable your soil is, or the slower it dissolves, conversely, the more stable that aggregate is. So this is an app I highly recommend. This is a really cool way to do it. I can send you the YouTube video where to find this at if anybody's interested in this too. Okay, any thoughts or questions on that? Actually, before I move into a more laboratory-based soil health measurement.
Do I need to start with a certain size of aggregate?
No, they say you don't. Yeah, it normalizes, so it just measures the area, whatever it sees. Like here, you can see how it kind of is working here. It uses image analysis software. It outlines your aggregate. You press start, and then as it dissolves, it will show you an aggregate stability score.
Nice.
Yeah, it's pretty cool. Any other questions or thoughts about this app? Did I sell anybody on this app? Is anybody going to go out and try it tomorrow after it's done snowing? Got a quiet group today. Maybe they're waiting until the end to hammer me with questions at the end.
They're sometimes a little quiet. I think they hope that I ask the right question. I mean, you sold me on the app. I think at our Nashua plots, we were trying some different cover crop and perennial ground covers. I'm excited to try that test out and see if there's differences during the growing season.
Yeah, that would be really cool. You have to let me know what the results look like. I'm curious if it does change over the growing season. You'd think it would with a cover crop system.
Yeah, it has to, right? They get different inputs at different times. I want to see if I can pick it up.
Yeah, that's cool. Yeah, that'd be a perfect undergrad project too. All right, so now I'm going to talk a little bit more about a test that you're all more familiar with. So this is the soil food web. At the base of the food web is the residue. So this is from a book on composting. So this might be soil organic matter, the crop residues that get input. So Dan was just talking about inputs. This is the energy and nutrients that come into the soil. Then there are these first level consumers that first colonize and start to break down the residue. These organisms over here we call shredders. They're larger. Many of them are insects or annelids, worms, that break the residue down into smaller pieces that the microorganisms like actinomycetes, fungi, and bacteria can break down even further and more efficiently. If it's in smaller pieces, increases the surface area. And then there are organisms that we often call mesofauna and protozoa, springtails, mites, columbella. So there are some mites that are pathogens to crops, but there are also a lot of good mites in the soil. Same with springtails and nematodes. A lot of the protozoa are important. One thing that's not featured here, but is kind of a frontier of soil biology and soil science, are viruses. Viruses are actually we think are really important for nutrient turnover in soils, but we don't really understand them at all. I think Dan and I need to talk our colleague Adina Howe into studying viruses in soils. And then up here is the tertiary or highest level consumers. One of my favorite critters is the Pseudo Scorpion. Actually looks like a scorpion. It has little pinchers. It doesn't have the stinger tail, and you can't see it with, well, you might be able to see it with your naked eye if it's large enough, but many of them you have to look at it under a microscope. Centipedes, other organisms. And so there are many tests. So the way that a healthy soil, one of my definitions of a healthy soil is fostering a diverse and rich population of these critters and all this biology, from microbes up to larger organisms like earthworms we can see with our eye. And there are many ways to measure their activity or their what they're doing in the soil or kind of assess them. One way is to measure the respiration and breathing, because many of these organisms are like us. They need carbon as a source of energy, and they also breathe in oxygen, and they release out CO2. So we can measure respiration or their breathing to measure their activity. We can measure decomposition. I talked about this measurement already, where we can bury something like tea or cotton and watch how fast it decomposes. We're kind of measuring the activity of these organisms at the base of the food web. And then we can just measure their overall abundance. So Dan and I have a colleague, Adina Howe, in his department, that she measures who's there in the soil. So what bacteria and fungi are actually there and their diversity. And this is a very, right now it's a moderately expensive and low throughput or not as easy to do on soils at the moment. But something that is easy to measure on the soil is the respiration, which is why I have this one in red. This is part of the CO2 burst, which is part of the Haney test. So we can measure these organisms, their breathing, and we think we can tell a lot of things about soil and nutrient cycling in soils in particular, just from their respiration. And so one way to do this, I mentioned this already, is measuring it with these Solvita paddles and color readers. They have actually other equipment now that can also measure this a little bit more accurately than these color change paddles. So the way these paddles work is that you put them inside the soil, you wet them up, and then the CO2 diffuses into these paddles. They actually start as a blue color, and the more yellow they turn, the more respiration is in. And so they show you here in parts per million how much respiration. They have a paddle reader that costs about $1,100, I think. You could stick your paddles in there, and it will give you a numeric value for how much respiration has happened in your soil. And that measurement, a lot of labs actually do, a lot of commercial labs will do, and it's part of the total Haney soil health test. And this came out of 3 papers, so 2 from Richard Haney, and using an extraction called the H3A. This is the authors, Haney, Hosner, and Arnold extract, and then Franz Lubers et al. Looking at the CO2 release in a 24-hour incubation, whether with Solvita or other measurements. And you come up with and so some of you that might have done this before, gotten a Haney soil health test on one of your field soils, this is how they calculate it. So they take the CO2 value, divide it by 10. The lower the number is in the denominator, the more weight or importance it has. Divide it by the water extractable organic carbon. So that one's divided by 100, divided by the water extractable organic nitrogen. Now those water extractable organic carbon and nitrogen is basically the food for the microorganisms in the soil. So it's a way to quickly extract what we think they can release and mineralize and make plant available over a year. And they, most of the labs, they'll give you a score that ranges from 0 to greater than 25. The greater the number, the more magnificent your mollisol is. The lower the number, according to the Haney soil health score, the more marginal your mollisol is. Okay, so I want to talk about the CO2 part of this test, and this is the last thing I'll end on. It's a pretty dense slide, but this is where I think we're going in that second objective. So where soil health tests might be able to inform management. And on the bottom here is the normalized CO2 burst value. And on the y-axis here is the economic optimum nitrogen rate. otherwise known as the EONR. This is how many pounds of nitrogen you need to add per acre to get corn to the agronomic optimum. And so there's a couple examples soils over here. So one soil B, had an EONR of 200 pounds of nitrogen. So at 0 pounds of nitrogen, the corn yield was only 50. You needed to add 200 to get to the economic optimum nitrogen rate. The soil A, however, had already very high nitrogen supplying capacity, and the more nitrogen you added, it actually went down. The yield was decreasing. There aren't very many soils like that, especially in Iowa, but there are two sets of data that collected soils from over 100 of these nitrogen rate trials. And I've combined them here together in this slide to show that the Franzluber 2018 data set, their 3-day incubation, Yost et al. Was done in the Midwest. Oh, I should mention that this Franzluber paper was in soils, Piedmont soils, and soils out in Virginia and North Carolina, whereas the Yost paper was a little closer to home. There were soils from Midwest states, including Iowa. And they did 2 different lengths of incubation, which is why I had to normalize the data. But when you overlay these 2 studies, this is where I see a promising trend, right? So these are the 2 studies overlaid, the Yost in the blue circles, Franzluber in the squares. And they show that the greater that CO2 burst test is, the lower the amount of nitrogen that you need to add to get to agronomic optimum nitrogen rate. And in the Franzluber paper in particular, this is actually a little bit both worrying and promising. There's a lot of soils that had a wide range of CO2 burst values, but had 0 optimum nitrogen rate. And I picked out just one of those soils over here, the soil A. But this shows you that there's something there. I think we need to understand where all this variability is coming from. But on average, for every about 10% increase in CO2 burst tests, it looks like there's a 24 pounds of nitrogen per acre to get to the optimum yield. So this to me is pretty promising evidence. And my lab group and others around the US and in other countries as well are looking at refining this so we can get to the point where we can make management recommendations based on this test. And at that time, that's all I've got. I've got a few more slides if we had time to talk about carbon, but I thought I'd end it there.
Perfect. Thank you so much, Marshall. I know I have a question or two, but I'd like to give other people a chance to ask some as well. So if you have a question, feel free to unmute and shout it out. Or if you are feeling a little shy, certainly feel willing and able to write them in the chat, and we'll make sure to read there as well.
Yeah, Marshall, Brian Doherty here. I was actually going to ask you about Alan Franzluber's work. So how far away are we from being able to use this for nitrogen recommendations, do you think?
Yeah, that's a great question. You know, at some point, I think we just have to say we've got enough evidence, let's give it a try. But I'm really hesitant to do that. And a lot of my extension colleagues, like John Sawyer, I know are too. There's a lot of variability here, right? This shows you a good example. So in these 2 studies, if you had kind of a 40% CO2 burst test, depending on how you measure it, you could have an economic optimum nitrogen rate of 0, meaning you don't have to add any nitrogen and your yield is doing pretty good. And you can have one that you need to add 200 pounds per acre. So I think we need some refining before, at least I wouldn't feel comfortable, you know, giving any farmer recommendations based on this yet. But I think this is where you can get into the nitty gritty. And what we're trying to do, we're trying to figure out what is explaining the variance from this relationship, and you know what's causing all these soils down here to read 0 but still have a wide range in CO2 burst values. You know, going back to a lot of those old nitrogen rate trial studies from the 80s, 90s, even the past couple decades, they had pretty noisy relationships between the LS&T test though and optimum yield for corn. So I don't know. I don't know. What are your thoughts, Brian? I mean, you're in this space too. You think we're ready to make recommendations?
Honestly, I think it's probably as good as some of the other recommendations out there, but I guess maybe the real question is, how do we find these soils that don't need any nitrogen? You know, where are they? Why don't they need it? And, you know, because we could save a lot of nitrogen application if we could pick out where those fields are.
You got it. That's the key. And my group published a paper last year where we tried to use kind of like a multi-phase test. So a measurement of nitrogen mineralization, a 2-week incubation, which is a little more onerous, and commercial labs won't do that, at least to my knowledge. But that helped at least parse out the responders and non-responders. So the ones you have to add nothing to, and then the ones that you need to add a little bit to. So that helped. You're right, but I think there are other soil properties maybe we need to know more about to figure out, at least between the ones we don't need to add any to. And there was actually only one soil from the Yoast paper that was in the Midwest that had a 0 EONR. And Sawyer was an author on this paper, and I meant to ask him what the deal was with that. I know a lot of these Franzluber, the squares down here, they either are corn coming out of alfalfa, which makes sense. Alfalfa is a really big nitrogen fixer, or they had animal manure. So bringing it back to the manure topic, they were getting slow release manure from the previous year, which is why they had a high CO2 burst, but also probably a low EONR, and didn't need any nitrogen that year. Great question. Any other thoughts or questions? I didn't see anything in the chat. Did you see anything in the chat, Dan?
I don't either. It is a quiet crowd today. One, thanks for joining us. I really appreciate it. And I also really enjoyed seeing the CO2 burst and your thoughts on where we could potentially go to or where we're trying to get. Certainly with Iowa, the nutrient reduction strategy and pressures on how to make manure plants better. That's a topic I think we'll continue to see interest in. I think it's extra nice that you pointed out that some of those more challenging soils might be soils with a manure history. I think that gives us something as manure management planners to think about if we would move towards a system like this. And then my actual question for you was about this slide as well. So they figured out the economic optimum after the season, right? And then you normalize that compared to the one they knew was optimum. How would we move towards preseason when we don't know what the optimum is going to be? Have to rely on a burst number telling us?
Yeah, you have to rely on the value. That's kind of the thing. It's almost like you're training the model, right? With that, you're right. The AONR usually is done, or EONR in this case. You do these nitrogen response curves. You collect the soil that year, and you're making this relationship between that year and the soil. But you want to predict next year's. And one thing that some of my colleagues are doing here is trying to use weather. We know weather affects this relationship too. And there's been study going back a couple decades from Larry Bundy and Dan Androsky, I think his last name is Androsky, from University of Wisconsin, that showed that, the LS&T test works great when there's certain weather conditions, but under certain other weather conditions, it doesn't. So, once we get to the point we can predict weather going out 6 months, at least, maybe we'll get there where we could use both the weather predictions and the soil test to inform this highly accurate measurement of how much nitrogen we need. There's also a debate going on now of whether we even need to be taking soil samples. Maybe we use remote sensing. It's not very feasible for a lot of growers to get out and sample in the spring, right? There's always already a tight timeline. Getting out there and getting a soil sample and sending that into a lab and then waiting to get the data back. Maybe we need to move to something like a Veris machine where you could drag it through the field and get some proxy for the nitrogen requirements for that particular part of the field too, right? Not every part of the field is created differently. There are going to be some parts of the field that need more nitrogen than others. And that also changes through years. So it gets really complicated.
Yeah, and your relationship here was more across fields rather than within fields, right? So it'd be interesting to see how much does that hold true within a field, right? For the economic optimum rate within a field.
Yeah, I would. I only know of one person. Maybe there's some older studies out there, but I know one person working on that right now, looking at kind of the spatial variability of the EO&R, or how much corn nitrogen needs vary across the field.
And then I think I have one last question for you, unless someone else types one in, and it's related to manure and potential carbon credits. Any thoughts on what adding manure to a rotation does for carbon storage, or how we would potentially claim some carbon credits from that activity? Is it
I'm
possible?
glad you asked me that, and I thought you might ask me that. I have a backup slide here on some carbon stuff. So this is where I would start. Somebody's done a meta-analysis on this already. There are some manure inputs that are exceedingly high, that are rates that people would never use, but they have both kind of the experimental data, and then I can't remember what these X's are. Dan, I think you and I have talked about this paper before, but this would be a place where I'd start, and it's about 20% of the carbon added from a manure, or not 20%, 10% of the manure ends up as kind of long-term carbon. So that could be a starting point if I were somebody trying to give carbon credits to people applying manure. Say about 10% of whatever the carbon content in that manure will give you that much carbon credit.
Perfect. I love that study personally. It makes trying to think about the system really interesting. And you were right, in the two high X's, they did add a lot of manure every year, but those studies were exceptionally long, if memory serves me correct, when they measured it. So they had a long-term cumulative effect, which was nice to see that it kept doing it for kept gaining carbon for a long, long time.
Yeah, and almost seems like, yeah, the more you add, you're getting kind of less gain.
Hmm.
You know, where here it's like a ratio of 10, right? But here you're getting to, you know, a little, no, it's more.
More.
Yeah, you're getting more than, yeah, so it's tilted up. It's not, it's yeah, more than 10, 12 or so 12%.
Which is interesting. I mean, I was with you. The more manure I add, eventually I'm going to get to this carbon equalization standpoint. And they said it was maybe the opposite of that for some reason. If I keep adding it, I get more and more improvement, and I just have to be a little patient about getting there.
Yeah, I mean, there is something to this carbon saturation idea, but I think, you know, our soils are about 50% of what they were prior to cultivation. So we've got a lot, and like, you know, a study we did, we showed that even after 40 years of converting cropland to CRP, so covered a lot of those soil health promoting practices, we didn't have the animals on there. I think that would have increased carbon even more. We could graze CRP, but we showed about it'd take about 100 years to get to native grassland. So about a 1% increase in soil organic matter, 1% of the whatever you start with, not a 1% of organic matter, but it'd take about 100 years to go from a 1% organic matter to 2%, or double it. If that
Yeah,
makes sense.
it does. Adding soil carbon is a long, slow process sometimes, which makes some of the other tests that you mentioned, the burst test, or maybe that slake test where we can assess structure, really interesting, where we can see some of the benefits shorter term.
Yeah, you got it.
All right. With that, a big thanks to Marshall. I loved hearing about soil health. It's a topic I don't know enough about, so especially nice to see that connected to manure. Thank you for joining us. Everyone else, thanks for joining us today. Hopefully we'll coax you into being a little more talkative at one of these in the future. I know as of right now, I haven't listed what our next plans are. I've worked out 3 out of the next 4 months, so do plan on us being back. And we appreciated having you today. So big thanks to Marshall. If you have any questions for him that you didn't get asked today, make sure you do reach out to him either on Twitter or via his email address. Hopefully he'll share that with me one more time, maybe in the chat. So
Yep, I'm
otherwise,
on it.
thanks, Marshall.